Hello my dear students, and welcome to the second life science lesson. In the first lesson, we have started chapter 1, which was control and coordination in living organisms, and we have discussed the first part of the chapter where we talked about stimulus, response. We've talked about how different plants respond in the form of movement, and we have also talked about the different types of movements in plants. In this lesson, we are going to talk about the second part of the chapter, which is response and chemical coordination in plants, with special reference to hormones. Here, we are going to look at how different plants coordinate different physiological processes within themselves in order to respond to different types of stimuli using special types of chemicals produced by the plants themselves known as hormones, or more specifically phytohormones. The word phyto here implies plants. So when one says phytohormones, it implies plant hormones. So now let us first look at the definition of phytohormones. In order to learn the definition of phytohormones, there are 4 points that you need to keep in mind. The first point being that they are organic chemicals. Phytohormones are chemicals produced within the plant themselves, and they are organic chemicals. Number 2, these organic chemicals are produced in small or minute quantities from special parts of the plant known as meristematic tissues. Now, you may recall from previous classes that meristematic tissues are the growing parts of the plant. And since phytohormones control growth, phytohormones will be specifically found in the meristematic tissues, or in short, meristems of these plants. Point number 3 is that after they are produced in the meristematic tissues, they have to be transported. They have to be taken from where they are produced to where they will perform their function. So they have to be transported. But how are they transported? They are transported in the sap via the xylem tissue. Now, as you may recall, xylem tissue is a part of the vascular tissue in plants. Xylem conducts the absorbed water and minerals from the roots to the leaves for food production. So, xylem will naturally contain sap. Sap is a mixture of the water and the nutrients that are absorbed by the roots. So, when this sap is being conducted via the xylem, these phytohormones are also conducted within the sap via the xylem. And lastly, once they are transported from where they are produced to where they need to be, there they will perform certain functions. So what is the basic function of a phytohormone? It is to coordinate or to manage different physiological processes. Now you may be wondering, what are physiological processes? Physiological processes are known as functional life processes. These are the processes that occur within plants without which the plant cannot survive. So therefore, physiological processes or functional life processes are known as life processes because they are essential for the survival of the plant. So what are these physiological processes that are being controlled by hormones? Here are some examples. Number 1 is budding. Budding means the predecessor to the flower. Budding is an essential life process which gives rise to the flowers. Now the flowers, they come about from buds. The flowers are known as the reproductive parts of the plant. Now without the flowers, reproduction cannot take place. Thus, it is a very vital life process. Next is the production and ripening of fruits. Now flowering will give rise to the flower. Once the flower performs reproduction, it forms the fruit. The ovary of the flower will form the fruit, and the fruit will give rise to the seeds. Now these seeds, in order to have new plants, new individuals, they need to germinate. Germination of seed is also a physiological process. Next, it also involves certain important processes such as cell division. Cell division is essential for the plant in order to grow and in order to reproduce, and also in order to repair and regenerate certain parts of the plant. So therefore, cell division is also another important functional life process or a physiological process, and cell division as a whole will lead to growth and development. Growth and development of the different parts of the plant, which brings about a holistic development and growth of the plant. Now all of these processes that I have just mentioned are crucial for the survival of the plant. That is why these processes are known as physiological processes. Now, it is very important to mention that phytohormones are also known as plant growth regulators. Why are they called regulators or growth regulators? Number one is because they influence growth, but more importantly, phytohormones or plant growth regulators, they either start a process or they stop a process. Or in other words, they either promote a process or they inhibit a process. Promote means to start, inhibit means to stop. So basically, phytohormones or plant growth regulators can be divided into two categories. The first category are known as promoters because they promote or they start different processes in plants. And this group of hormone comprises of auxin, gibberellin and cytokinin. The other group, which are known as inhibitors because they stop or inhibit a process, include different hormones which are ethylene and abscisic acid. But for the purpose of your syllabus, we are going to only discuss about auxins, gibberellins and cytokinins, which belong to a group known as the promoters. Now we are going to look at the role of different phytohormones. What role do they play in plants? Now firstly, let us look at what are their roles. As I've already discussed before, the main role of phytohormones is to coordinate different physiological processes. Now, what are these physiological processes? I have also already discussed them earlier. Now here you can also have a look at them. They control apical and lateral growth. Apical meaning the growth of the apical part of the plant or the apex of the plant, the tip of the shoot and the tip of the root. Lateral growth means growth laterally or sideways. It also controls budding. Budding leads to flowering. Flowering produces flowers, but after pollination and fertilization within the flower, it leads to the formation of fruits. Now from the fruit you get seeds, and seeds will germinate. So germination of seeds are also controlled by phytohormones. They also control different tropic movements, which we have discussed in the previous lesson, and they also control ripening of fruits and other processes. They function in small quantities, which we already know from the definition itself, and they, as I've already told you, they either promote or they inhibit a process, meaning to say that they either start a process or they stop a process. Now let us look at the source. Where will you find phytohormones? Again, I've already told you phytohormones originate from special parts of the plant or the growing parts of the plant known as meristems or meristematic tissues. You will find the meristematic tissue in the growing parts of the plant, which are young leaves, buds, seeds, fruits, etc. Transport. Now, how are phytohormones transported from where they are produced to where they need to control these different physiological processes? They are transported, as I've already talked about earlier in the definition, via sap through the xylem primarily. And lastly, what happens to the phytohormones after their use? They are destroyed after use since they cannot be stored. So, phytohormones need to be continuously produced in the plant. Now, moving on to the next slide, we are going to talk about the different types of phytohormones. As I've already told you earlier, we are only going to discuss the first group known as the promoters, auxin, gibberellin and cytokinin. They are called promoters because they start a process. So the first hormone that we are going to look at is auxin. Now auxin is the first plant hormone. It was first discovered by a person known as Charles Darwin in 1880 from the cotyledons of growing grass. It was named as auxin by another scientist called Wendt in 1923, and in 1934 it was isolated and studied separately by another scientist known as Skogel. The origin, where will you find auxins? Auxins will be primarily found in the apical meristem. The word apical implies the word apex. which means the tip. So you will find auxins on the tip or within the tips of the shoot and the root. So you will find it in the apical meristem, you will find it in young leaves, in fruits, and also in roots. Now let us look at the functions, the most important part. Let us look at the functions of auxin. Number one, it promotes apical dominance. Now in order to understand apical dominance, first we have to understand that plants have different types of buds on its body. The buds that are present on the tips are known as apical buds because they are present at the apex, and the other buds which are present on the sides of the plant are known as lateral buds. Now when apical dominance occurs, apical dominance is a phenomenon by which the growth of the apical bud inhibits or hinders the growth and development of the lateral buds, which means to say that the growth of the apical bud takes place faster than the growth of the lateral buds. So this apical dominance is brought about by auxin. So whenever you hear the word auxin, auxin is always associated with apical dominance. And a better way to remember it is A for auxins, A for apical dominance. Number two, since it controls growth, it controls cell division, which brings about growth. So cell division is controlled by auxin, and after division, the resultant daughter cells are also enlarged. That means the whole size of the daughter cells also increases because of auxins. Next is the growth of roots, growth of fruits, and it also promotes a certain process known as parthenocarpy. Now what is parthenocarpy? Parthenocarpy is the formation of seedless fruits, fruits without seeds, without pollination or fertilization. So naturally, whenever there is a flower on a plant, whenever there is pollination, which is the transfer of pollen grains, pollination leads to fertilization, which is the union of the male gamete, which is the pollen grain, with the female gamete, which is the ovule. Now the pollination leads to fertilization. During fertilization, fusion of the male and female gamete takes place, which leads to the ovule turning into the seed and the ovary turning into the fruit. So naturally you will have a fruit within which you will find numerous seeds. But parthenocarpy is an exception where there is formation of fruit without pollination or fertilization. And since there is no fertilization, it does not involve the fusion of the gametes, hence the ovules will not turn into seeds. So parthenocarpy leads to the formation of a fruit which is seedless, and that parthenocarpy is induced by auxins naturally. Next is it controls tropic movements. As we've discussed in the previous lesson, tropic movements are those where the direction of the stimulus influences the direction of the response. Tropic movements are also controlled by auxins. Now this diagram will demonstrate how auxin influences different types of tropic movements. So the first type of tropic movement is phototropic movement, where the plant responds positively to light. The stem of the plant responds positively to light because it moves towards the light. Now here in this diagram, as you can see, this is a potted plant which is kept in a dark room with only one source of light, which is this torchlight. Now the light is coming from the right hand side, so naturally the left hand side of the plant will be in shadow. It will be a shaded portion. So now the auxins will develop and they will be transported to the portion which is shaded, to the shaded portion of the plant. Now when auxins start accumulating on the shaded portion of the plant, higher concentration of auxin stimulates cell division. Cell division leads to growth. So here cell division and growth is taking place in the shaded portion of the plant, which will grow more compared to the unshaded portion which is receiving light. Therefore, if this part grows more than this part, if the shaded portion grows more than the unshaded portion, naturally the stem will start bending towards the light, and this is known as phototropic curvature, and this is brought about by higher concentration of auxin. So in this way, auxin controls phototropic movement. Now in order to see how auxins control geotropic movement, we have to look at a seed that is planted in soil. Now a seed, once it is planted in soil and it comes across favorable conditions like water, it will start germinating. When a seed germinates, its embryo gives rise to two parts, which are plumule, which gives rise to the shoot, and radicle, which gives rise to the root. Now the plumule has to bend upwards in order to come out of the soil and give rise to the root, whereas the radicle has to grow downwards into the soil in order to form the roots. Now here, with regard to plumule, again, auxins will be deposited on the shaded portion or the lower portion. Now, I have already told you earlier, higher concentration of auxin leads to more cell division and more growth. So naturally, if the shaded portion of the plant of the plumule grows more than the unshaded portion, it will lead to this plumule bending upwards and coming out of the soil. Similarly, if you look at the radicle in order to give rise to the root needs to bend downwards. So here, auxin will again not be deposited on the upper side, but again it will be deposited on the lower side, because here we need to understand that the root behaves differently compared to the shoot with regard to auxin. Here, the auxin will be deposited on the lower side again, but in the root, higher concentration of auxin inhibits cell division. So therefore, here, as opposed to the plumule, cell division will take place more on the unshaded portion. And when cell division takes place more on the unshaded portion, it leads to the radicle bending downwards into the soil, giving rise to the root. So we have discussed about the first phytohormone, which was auxin, and we looked at its different functions. Now we are going to look at the second phytohormone, which is known as gibberellins, or some people even call it gibberellins. Now, gibberellins was discovered in Japan by a Japanese scientist whose name was Kurosawa in the year 1926 from a special fungus named Gibberella fujikoroi. This fungus, Gibberella fujikoroi, was growing in paddy plants, paddy meaning rice plants, and this was causing the rice plants to grow very, very tall, unnaturally tall. So Kurosawa discovered this hormone from this fungus known as Gibberella fujikoroi, which was growing in paddy plants. The name gibberellin was given by another scientist whose name was Yabuta in 1935. And today, over 100 varieties of gibberellins from different plants are known to man. Now, gibberellins. Where will you find gibberellins? The primary place where you will find gibberellins is in germinating seeds. A very easy way to remember this is G for gibberellins, G for germinating seedlings. So gibberellins, you will find it in germinating seedlings, you will find it in mature seeds and growing leaves. Now let us look at, most importantly, the functions of gibberellins. Number one is it breaks seed dormancy. Now dormant or dormancy implies inactivity. A seed remains inactive when it has not come across favorable conditions like soil and water. If there is no soil and no water, the seed remains inactive. But however, as you expose the seed to favorable conditions, it will become active, thus breaking its dormancy. And this breaking of dormancy is done with the help of gibberellins. Now, after breaking seed dormancy, it also affects cell division, which leads to the growth and germination of the seed. But here, the newly divided cells become longer, they elongate, their length increases. So, cell elongation is also affected by gibberellins. If individual cells of the plant become longer, eventually the whole stem of the plant will become longer. So, gibberellins also leads to elongation of the stem. It also influences fruit growth or growth of the different types of fruits that you can see in the market. And lastly, like auxins, gibberellins also induces parthenocarpy, which we've already talked about earlier, which is the production of seedless fruits without pollination or fertilization. My dear students, we have already talked about two phytohormones, which were auxins and gibberellins. Now we are going to talk about the third type of phytohormones, which is cytokinin. Now, in order to understand cytokinin, let us look at its history. In 1955, a scientist called Miller, with the surname Miller, discovered a substance in yeast which is a unicellular fungus, and he called that substance kinetin. Similarly, in 1963, another scientist whose name was Latham discovered a similar substance in maize, and he called it zeatin. Now, similar substances like kinetin and zeatin were discovered by different scientists in different plants, and all of these substances seem to control growth or perhaps cell division at a particular stage known as cytokinesis. Thus, this group of hormones was named as cytokinin. Again, it was named cytokinin because it controls cell division at the particular stage known as cytokinesis. Now, let us look at where you will find cytokinin in plants. In plants, cytokinin is found in roots, it is found in leaves, and it is found in the endosperm of seeds. Endosperm meaning the reserve food material for the growing embryo. Now, endosperm, you will find it in coconut milk or coconut water, tomato juice, apple fruit extract, and maize kernels. Now, a simple way to know the endosperm is we all eat popcorn, isn't it? So, when you put the corn into the utensil in order to cook it, it pops and there's a white substance that comes out, isn't it? So, that fluffy white substance is actually the endosperm. Now, let us look at the functions of cytokinins. Now, the function of cytokinin is it controls or it influences the growth of the lateral buds, opposite to that of apical dominance. Earlier we saw with regard to auxins, it induces apical dominance, where the apical bud inhibits the development of the lateral buds. But here, cytokinin promotes the growth of the lateral buds. Number 2, it delays senescence. Senescence meaning aging. As time progresses, we all age. Human beings, animals, plants, we all age. So naturally, aging brings about wear and tear. And wear and tear can cause some parts of the plant to fall off. For example, leaves, they fall off during autumn. Or when the fruits mature or they ripen, they fall off the plant. That is a symptom, or that is a characteristic of senescence, aging. So cytokinin delays the process of aging or delays senescence. And lastly, it also promotes seed germination like gibberellins. So my dear students, we have seen 3 different phytohormones in plants that are produced naturally. Now we are going to discuss about artificial or synthetic phytohormones, which means that these artificial or synthetic phytohormones are made by man artificially in laboratories, and they allow us to manipulate plants to use them for our benefit, for let's say commercial benefit, for the production of fruits or for the production of flowers, or using these phytohormones to induce growth in certain types of plants. So here you will see different types of phytohormones. These are the names of different types of artificial or synthetic phytohormones. Synthetic auxin, synthetic gibberellin, we have IAA, which stands for indole acetic acid, IBA, which stands for indole butyric acid, IPA, which stands for indole propionic acid, and NAA, naphthalene acetic acid. So these are the names of some of the different artificial or synthetic phytohormones. Now let us look at the different uses of synthetic and artificial phytohormones. So here are a number of uses. So primarily we can use phytohormones, artificial phytohormones, to induce the rooting in different types of cuttings. Now, as you may know, there are certain plants that have the ability to be grown vegetatively. By cutting a vegetative part of the plant and planting it elsewhere, we are able to get a completely new plant from that part that was cut. Now, the part that was cut from the original plant to be planted elsewhere is known as the cutting. Now, when we apply certain artificial phytohormones on the cut part of that stem, it speeds up the process of rooting, and it allows that cutting to initiate rooting at a faster rate, and also a larger volume of roots can be seen coming out from the cutting. So that is the first and primary function of phytohormones, in order to induce rooting in cuttings. Number 2 is fruiting or parthenogenesis. So we may also use different phytohormones to induce fruiting, say to produce fruits out of season, or to produce fruits without seeds, parthenocarpic fruits, where the fruits do not have seeds because the pollination and fertilization has not taken place. For example, you can see different fruits in the market like bananas or grapes which do not have seeds inside them. So these are all parthenocarpic fruits which have been produced by using different phytohormones to induce parthenogenesis. Number 3 is delay of pre-harvest fruit crop. So in nature, naturally what happens is the plants bear fruits, and sometimes these fruits fall off the plant before ripening or before they harvest. Now this affects the yield, the fruit yield for the person who is growing those fruits. So in order to delay the falling off of these fruits or vegetables from these plants, different farmers can use phytohormones to delay the pre-harvest fruits. Next is herbicides. Herbicides are certain chemicals which are used to kill unwanted herbs and plants that are growing in your garden or your farm. So instead of manually removing these herbs or shrubs, we can use phytohormones. We can apply phytohormones which does not allow the growth of these plants, and thus we can use them also as herbicides. And lastly, healing wounds. Now plants also get induced with certain injuries sometimes. Sometimes there are certain animals that enter the farm or the garden and they inflict certain injuries on these plants. Now if these injuries are not dealt with, they will damage the plant, and the damage to the plant will result in maybe decrease in the yield of the fruit or the vegetable. So in order to heal these wounds as well, we can use phytohormones which induce cell division and cell growth, which heals the wound of the plant, making the plant healthy. So with that, we have come to the end of the second part of the first chapter. We have just discussed about how plants respond to different types of stimuli by coordinating different physiological processes with the help of special chemicals known as phytohormones. We have looked at 3 different phytohormones, which were auxin, gibberellin and cytokinin, and we also talked about certain different artificial or synthetic phytohormones and their uses. Now in the next lesson, we will be discussing response and chemical coordination in animals with regard to animal hormones. So thank you for listening, and I will see you in the next lesson.